again, at any time, feel free to stop me, ask questions. I'll try to work things out for you, and just don't be afraid to speak up and tell me if I'm like giving too much information or anything. And I was gonna say something else, but I forgot. So anyways, going into the lecture for today. So biotechnology really deals with cells and disease. So diseases are, well, I'm sure you guys know what diseases are, but it's when our bodily functions don't operate normally as they should, resulting in health complications and just messing up our internal functions and parts of our bodies. So there are 4 main kinds of diseases. There are infectious diseases, deficiency diseases, hereditary diseases, and physiological diseases. So going through each of them, can anyone give me an example of an infectious disease? So like a hint would be infectious. Okay, yeah, the flu. So the flu can spread from person to person, and that's how we know it's infectious. Not 100% sure, actually. Yes, that's definitely, that's an example. But yeah, so infectious diseases are able to spread from person to person through various agents like water, the air, etc. Deficiency diseases are when your body lacks something. So for example, you could have a iron deficiency, and that could lead to complications. So today we're going to mostly be looking at hereditary diseases, which deals with genetics and how cells divide and pass on genes to one another. In cell biology, we're going to be mostly focusing on that. Physiological diseases are just like things that deal with your body and anatomy. So like if you broke a bone and that led to something, cancer, high blood pressure. Yes. Yeah, exactly. Those would be examples of physiological diseases, I think. So, don't quote me on any of this, because the only thing I can tell you for sure is the hereditary disease one. But yeah. So basically, in order to try and solve disease and such, we have to look at cells, which are, again, the building blocks of life. So using cells, we can figure out the best way to treat a disease based on how your body is going to react. So a lot of diseases deal with the cell cycle, which we looked at last week. And so an example of a disease that is regulated by the cell cycle is cancer. And this is when cells grow out of control. So as you can see, there's a very direct correlation between the operation of cells and also the formation of this disorder. And other examples of how cells play in is, again, DNA and passing on genes. So if you remember from our second class, I believe, or no, first class, well, first and second, DNA is the book transcript of the cell. It codes, it has all the information for the genes. And so when you get like color blindness from your mom and your dad, you have to figure out how your DNA is like passing on genes so that you get color blindness, and then we can like interfere based on the kind of disease that it is. And also another example of cells would be in a physiological disease. For example, if you like scraped your knee and then your body was trying to like replenish the skin where you lost where the wounds cut open, platelets would come and stop the blood from flowing out, and platelets are a specific type of cell. So again, everything just basically has this connection to cells, and they are very important, but for now, we're going to be focusing on their DNA and genetic material. So what actually is biotechnology? The the direct definition of biotechnology is the manipulation of biological systems to create products. So this means exploiting and using biological organisms to create certain things that will help humanity and our bodies. So for example, bacteria is often used because bacteria has these genes called plasmids that we'll look at in the next slide, but bacterial genes are used to transfer certain genes from like one entity to another to make like human growth hormone. So humans have something called a growth factor that, true to its name, helps you grow and develop, but some percent of the population, they don't have genes that code for this growth factor, so they need this growth factor from other sources. And humans have used biotechnology to exploit this in cows. So some cows create like hundreds of thousands of times of this growth factor in their milk. And so if people grow or if people drink this milk, then they'll get the growth factor. And that's how you kind of can engineer the cow. Oh, well, I guess engineering the cow would be cloning this cow so that it produces more growth factors. But yeah, that's an example of biotechnology. Yeah, cow cloning. So you have a cow that has a lot of this thing that humans want, but there's only one cow. So if you want a bunch of cows that have a lot of this thing, you need to clone the first cow with a lot of this thing. So you have a bunch of cows with a lot of this things. And that's how you use biotechnology. Cloning is biotechnology. So there are four main uses of biotechnology: medicinal, which is our focus. This includes vaccine, insulin, growth hormone, which I just talked about, diagnosis, so diagnosing certain diseases, and also gene therapy. Okay, so you can breed cows, but it's a very risky process, because when you breed cows, you're not sure that this gene will definitely get passed on. Just like if your mom has like blonde hair, it's not for certain that you're going to have blonde hair. So it's much less like risky to clone a cow and its genes than it is to like breed this cow, but it also requires very advanced technology, which is why most people just breed cows. But then that wouldn't be biotechnology because we're not manipulating the system. It's just artificial breeding and selection, which we don't really do anything except put cows like together. If that makes sense. It's easier and it's more cost efficient, but it's less like certain. You just never cut your hair if you... What? What do you mean, Eleanor? Wait, I lost the chat.
Like if you cut your hair, it's gonna be short because, yeah.
Wait, where are you getting like the cutting your hair from?
Like if your mom has long hair, well, you can basically just cut it to make your hair short. So like,
okay, I meant like blonde hair, so like the color of your hair.
oh, I thought
Yeah.
I heard.
Yeah, okay. I was confused. What you said makes sense though in your context. But yeah, so biotechnology is used in medicine, industry, food. I'm sure you guys have all heard of genetically modified organisms, and below the diagram on the right is a list of all of the genetically modified organisms so that they can become bigger and more tasty. And yeah, and in the environment, we can use biotechnology on a process called bioremediation, which is utilizing technology to solve some aspect of the environment. For example, if you have an oil spill, you can use bacteria to like slurp up the oil. All right, biotechnology in medicine. Again, it is used to prevent, so stop the happening of, diagnose, and treat illnesses. So these are the three aspects, and it can also be used to improve medicine because of the pieces of knowledge it provides. So an example of a biotechnological technique we're going to look at is PCR, which is amplifying DNA like billions of times. And so this way we have a lot of DNA that we can study. And so we can also use something called Sanger sequencing, which is sequencing DNA. So we can see like what the information that the DNA has actually says. So using biotechnology, we can study again how the human genome is sequenced, how genes are passed down, and also how DNA can be repaired, because we know like how DNA is sequenced. So there are many different kinds of medicinal biotechnology fields. So we have genomics, dealing with the genome, pharmaceuticals, dealing with drugs, DNA sequencing, also dealing with the genome. So I don't know why that's on there twice. Cell culture, which is an example of the cow cloning that we talked about. You would raise more cells to have this certain factor, artificially, of course. Interference RNA, which is something that we don't need to know about. And the genome, again, man, I put a lot of genomes on there. Okay, anyway, so these are a bunch of different methods that you can use. You have gene sequencing, which is determining the sequence of DNA, tissue engineering, which is just changing tissues in the body to have more or less of a certain thing. Antibiotics are an example of biotechnology utilized by, or that involves the bacteria penicillin, or not all antibiotics deal with penicillin, but that's the first one. If you cut, so basically you raise cells so the cells can reproduce with the okay, wait. Okay. So gonna go back to the whole thing. So you have this cow, right? This cow.
Yeah.
This cow cells have a lot of, let's say, A. So A is this thing that cells have, or cow cells have a lot of. And humans want A. But when you have a cow with A breeding with a cow without A, because A is rare, so without A is more common. But when you breed them together, you don't get A out. So what do you need to do? You need to take the cow's cells from A, and you need to take their cells and you need to make new cells. So cell culture is like growing cells in a lab. So you can take this cow with A's cells, and their cells will also have A. And if you can clone that cell and grow more cells with A, that's like a surefire way to get more A. Does that make sense? Like there's no like reproduction involved because it's like cloning. There's no other parent. So they goes in labs making things illogical. So they grow cows in labs making things illogical?
Like I feel like I don't understand.
Okay. Yeah, so basically they do grow cows in labs. If a cow has something that humans really want, they'll take the cow and they'll clone it in a lab. So also it's not like the whole cow, it's only the cells of the cows.
Wait, but what do they do with the cells?
They grow more cells. Okay, so they grow more cells and then they take those cells and they can put it into a new cow. And then the new cow now has the cells from the old cow, and then the new cow becomes like the old cow.
Oh, okay.
Does that make sense?
It makes more sense than before.
Cowception. Yeah. Yeah, and then they just repeat the process over and over again. Cow fusion.
Ladies and gentlemen, today we'll be dissecting a cow live. We're going to take out the cell and then clone it.
Yeah, I actually got my cow here. No, my cow. Yeah, there's no cow. Okay, anyway, but yeah, so it deals with certain parts of the cow and not the entire cow. And it could also be another organism, not just a cow. So this is just a timeline for biotechnology and what different techniques and products have been created. I am not going to go over anything of this. This is just if you want to see this slide after class, I'll put this into Google Classroom. But okay. I guess I'll capitalize on some important things. So 1953, structure of DNA is created, double helix, DNA polymerase. Actually, you don't need any of this. Never mind. All right, recombinant DNA. So what is recombinant DNA? It is DNA from 2 or more hosts. So this means that you have one piece of DNA, but then it has DNA from 2 hosts. So for example, if a piece of DNA has bacterial genes and human genes, then it's recombinant. And so recombinant DNA is formed in 3 ways. And so the first way is transformation, which is selecting the gene that you want. You cut it with restriction enzymes, which are basically molecular scissors, and then you attach it to something else like a plasmid or a virus. A plasmid is a bacterial chromosome which has DNA in it, and it's circular. And plasmids are often studied in biotechnology just because they're so versatile and they don't affect the genome of the bacteria. They're just this floating thing. And then you also add a selectable marker. So for example, if you have a green marker and then the green shows up into the host cell, then you know it worked. So an example of this would be if you have a bacteria that is resistant to heat. You cut that heat resistant gene, put it on a virus, and then the virus goes into a human cell. And then suddenly the human cell is also heat resistant. That's how you know it works. Non-bacterial transformation is just bacteria, or transformation with no bacteria. Phage induction is that instead of bacteria, bacteriophage is used, which is a virus that attacks bacteria. Kind of confusing, I know. But recombinant DNA has a lot of uses. For example, if you have a crop that has a gene that means that makes it drought resistant, and you want more of your crops to be drought resistant, then you can take this gene and add it into other entities, beings, vaccines, prevention, etc., etc. All right. Let me see where we are with time. Okay. I think we're good with time. Shoot, what did I do? How do I go back to present? All right, there we go. So the first technique we're going to be looking at today is something called gel electrophoresis. So gel electrophoresis is a biotechnology technique that just basically separates things based on their size. And so usually it's going to be a mixture of DNA and RNA. So RNA is just, if you want to make a protein, you have to first have your DNA with that codes it. You have your RNA that takes the DNA code and gives it to the protein, and then you have your protein. In case you forgot what RNA was. So basically all of these molecules are put on this sheet thing, as you see in the bottom left. I did not forget my right and left. And then so you put them into the small little containers, and then an electric field is pushed. And so on the other end, it's positive. On the upper end, it's negative. And then so this field pushes the molecules down because of the attraction. So most DNA, RNA, and protein are negatively charged. So they will be pulled down towards positive, and the smaller your molecules are, the further it'll travel. You do not need to know about sodium dodecyl sulfate. But yeah, just remember that gel electrophoresis separates things based on their size, and the smaller ones travel the furthest. Blotting. Okay. Blotting is like my favorite kind of biotechnology, and it's used for identifying specific biomolecules in a mixture. So if you have like a mixture full of DNA, RNA, and protein, and you're just trying to look at DNA, then blotting is the way to go. So first, a particular type of molecule is extracted and then treated to be run through a field, much like what we saw in gel electrophoresis. There's a field, and then the proteins are denatured, the disulfide bridges are broken, and restriction enzymes, so the snippy things, are added to nucleic acids. So this just means that before you run it through the field, you have to break it down. And then gel electrophoresis is used. So the molecules are separated based on size, and the negative molecules will get trapped while the positive ones travel further. And then we use a selectable marker, again, something that tells us that the molecule is there. These are detected using a probe that helps you visualize your targeted molecule. So there are many different kinds of blotting. Northern blotting deals with RNA. Eastern blotting deals with something that's completely different from the other kinds. Western blotting deals with proteins, and Southern blotting deals with DNA. So there's a lot of different kinds of blotting, and they're all named after directions for some reason. But let me see how many slides we have left. Oh, geez. Okay, we have a lot of slides left. I'm just going to like speed run through them. All right, so sequencing. Sequencing is basically determining the order of a nucleotide sequence in a DNA. So this is useful for identifying mutations, because if you have one nucleotide that isn't supposed to be there, that is there, you can look at it immediately because you have the entire sequence sequenced. So it's really easy to see when something is different when something's different than what it's supposed to be, and then you can immediately treat it. So Sanger sequencing is one type of sequencing, and it's useful if you have 900 or less base pairs of DNA, and it was used in the Human Genome Project. The Human Genome Project was started in 1993 and completed in 2003, and it dealt with sequencing the entire human genome. So all of our genes were sequenced. So there are 3 steps. First, DNA is denatured, so that means it's broken down. Then the temperature is raised, and DNA polymerase adds nucleotides. And then, so this creates a lot of different strands of DNA that can be separated using gel electrophoresis again. Next generation sequencing is the new kind of sequencing, and it's a lot more cheaper, cost efficient, deals with smaller reactions, etc., etc. So basically, it's just a lot better, because as technology goes on over time, it improves. PCR is like the biggest biotechnology technique besides CRISPR-Cas9. So PCR, also known for polymerase chain reaction, is amplifying billions of DNA, making billions of copies in a very short amount of time. And for that reason, its nickname is molecular photocopying. And then the inventor was credited with a Nobel Prize in chemistry. So there are 3 steps: denaturation, DNA is broken down; annihilation, primers attached to DNA, and then you start to get DNA synthesis, elongation. You get different kinds of DNA. You get like different strands of DNA. So although it may sound just like another amplification technique, the thing is, it's very, very fast. In 1 minute, 1,000 base pairs can be copies. So it's like, it can go to billions of copies of DNA in very little time. And it was also used in the Human Genome Project, used in DNA fingerprinting, which is a technique used in forensics. And yeah, so I like got into a fight with my friend over this like a while ago, but like basically all of biology is just the application of chemistry. So like when you get to like higher education and like college and stuff, like all of biology is just like calcified under chemistry. I don't know. It's like whack. And then chemistry is just the application of physics. Oh, because there's not like a specific Nobel Prize for like biology. But like, yeah. Anyway, DNA fingerprinting. Let's see. I gotta selectively. All right, mini satellites. So you know how we share like 99.6% of our genome with like apes? So that's 0.4%. So basically what I'm trying to get at here is my genome and your genome are like kind of the same, but then for the most part, but then there are these pieces of DNA that vary a lot from person to person. And it's these pieces of DNA that like define us. And so DNA fingerprinting targets these pieces of DNA called mini satellites. And basically, it's like a DNA fingerprint. The chances of two people having the same mini satellite sequence is very, very small. So at a crime scene, if a criminal leaves behind a tiny piece of DNA, then we can identify the mini satellites and compare all of the suspects. And so looking at the diagram on the right, you have the crime scene. You use DNA fingerprinting to sequence all the mini satellites, and then you map it to each person, and you can see such a suspect 2 is the culprit. Yes, kind of sus. I agree.
But what if this suspect is super sneaky and it puts someone out and somehow gets the suspect's fingerprint on there?
Well, I mean, that's just kind of always like a possibility in forensics. So you just gotta trust the technology, you know? All right, how many slides? All right, not that many slides actually. All right, stem cell research, gonna go through this real fast. So stem cells are cells in the body that can differentiate into any kind of cell. So for example, your nerve cells cannot turn into blood cells, but your embryo cells, when you are a fetus, they can turn into a blood cell or a nerve cell. So it's like certain cells can turn into other cells, but certain other cells can't turn into other cells, and stem cells can turn into any kind of cell. Different uses for stem cells. You can study how diseases form. You can study regenerative medicine, which is just basically like, if you break a bone, how do we get your bone back? You can also use it to test new medicine safety, so seeing how a cell reacts to it. And stem cell research is used in clothing. And for example, if you have our DNA, you take out the DNA, you put it into a fetus, and then it turns into another you. So a pluripotent, so going back to what I was talking about with like cells being able to turn into any different kinds of cells. So this is called totipotency, and totipotency is a measure of how, I'm answering a question, by the way. So someone asked, what is a pluripotent cell? So totipotency is the potential for a cell to turn into other cells. So stem cells are called totipotent. This means they can turn into every type of cell. Pluripotent cells can form every kind of cell except for new embryos. And then it's like, I forgot the ones after pluripotent actually, but it just goes like increasingly down in the potential to form different cells. Scientists have used stem cells to bring an extinct organism back to life before. That is really astonishing, but yeah. Gene therapy. Gene therapy is just introducing a certain gene into patients to cure a disease, and it's highly controversial in terms of ethics because it's really dangerous. Like if you take a foreign gene and you put it into your body, you don't know how your body is going to react. So it's only tested on disease that have no cure, and it could possibly cure cancer, AIDS, and cystic fibrosis. CRISPR is most notorious, and it's basically just gene editing. Target mutations, cut genes, blah, blah, blah. Ethical concerns. Obviously, biotechnology is not a light topic, and it holds a lot of weight because you're really messing with like a person's natural body. And so people have debated whether or not it should be allowed. Is CRISPR edible? I mean, I guess you can eat it. It's just protein. So like, yeah. But basically, some people don't like biotechnology. Some people like biotechnology. Let's go. All right.